Hello, I'm Michael Dustin from the University of Oxford and New York University School of Medicine. Welcome to part 3 of the Immunological Synapse, focusing on extracellular vesicles. This is an observation that we've made recently in the interface between the T lymphocyte and antigen cell during this critical communication that integrates innate and adaptive immunity. So the communication between the T cell and dendritic cell takes place through recognition of T cell receptors on the surface of the T cell with the MHC peptide complexes on the surface of the dendritic cell, assisted by adhesion molecules like LFA1 interacting with its ligand ICAM1. This recognition process generates a number of signaling outputs in the T cell and also feeds back information to the dendritic cells or other types of antigen cells, such as macrophages for killing microbes or B cells for production of antibodies. Experimentally, we can recapitulate some aspects of this communication by replacing the antigen cell with a supported planar bilayer, supported lipid bilayer containing purified adhesion ligands such as ICAM1 and the MHC peptide complex in a laterally mobile form. The lipid film in this case is in a liquid disordered state. Transmembrane proteins in this system become immobile, but if you anchor the proteins to the lipid bilayer directly, for example, through C-terminal histidine tag and some of the protein extracellular domains linked to nickel chelating lipids in the bilayer, the ligands are highly mobile and allow formation of an immunological synapse. So we can use a quite varied microscopy toolkit to interrogate these systems. And for the supported planar bilayer system, we can use wide field fluorescence microscopy when we're focusing on molecules that are in the bilayer itself, since molecules that are in the cell would be out of focus at the interface and interfere with that, with imaging of things in the interface. We can, in those contexts, switch to using total internal reflection fluorescence microscopy, which generates a very shallow evanescent wave at the point where a laser beam is reflected off of the interface. So this is a technology that became available around 2000 in a commercial form, very high numerical aperture objectives, and we use it very widely. It's also allows single molecule imaging in the context of a cell bilayer interface, which is a methodology I'll describe later. We also make extensive use of reflection interference contrast microscopy, which is based on interference between light that's reflected off of the interface between the cover slip and the media, with interference, with reflections that are coming off of the interface between the media and the cell. So those different reflections are phase shifted. So when the cell gets very close to the substrate, say within 100 nanometers, you start getting destructive interference. So the closer the cell becomes to the substrate, the darker the image will appear. That's very useful for looking at contact. In this talk, I'll also highlight the correlation of various types of fluorescence microscopy with electron microscopy, and particularly with electron tomography, which is a method where you take relatively thick sections for transmission electron microscopy. Say routinely, you might use sections that are a few nanometers. In the case of electron tomography, you use sections that are maybe up to 100 nanometers in thickness, stained similarly for other transmission electron microscopy with heavy metals, but tilted in the electron beam so that you collect a data set which can be used to deconvolve a three-dimensional image, again with very high sub-nanometer resolution. So this is a powerful method for looking at structures in the interface. Doesn't necessarily give you molecular identity, but that can come from correlation with fluorescence microscopy. So that's pretty much the toolkit that I'll be using for the studies shown here. So the story I'm going to tell you about extracellular vesicles enriched with T cell receptors, we started seeing indications of this phenomena about 20 years ago now, when we first started looking at T cells interacting with these planar bilayer substrates. And this is an example from a collaboration with Emil and Anaway. We were in St. Louis, where we're basically looking at a T cell forming an adhesion based on the interference reflection microscopy, again, where the contact areas appear dark, and the accumulation of T-cell receptor in this interface. The substrate in this case is coated with purified MHC peptide complexes. So this is a purely T-cell receptor-driven adhesion. Now, this is not the way we think things happen physiologically, but as an experimental model, we were kind of interested in how this would, basically what the capabilities of the T-cell receptor MHC peptide complex are, and this allowed us to basically see that the T-cell receptor would cluster in this interface if you had sufficient densities of MHC peptide complexes. However, something that surprised us is that when we removed the cell by just basically flowing media over the surface where the cell was adhering, we actually found that we could knock the cell off, but would leave a set of contact areas, and that these were positive for the T-cell receptor. So we were somewhat surprised to find that the T-cell would so easily release this T-cell receptor, which everyone had assuming would be internalized and degraded following its interaction with the MHC peptide complex. But this gave us an indication that T-cells could in some way release their T-cell receptors towards a antigen-presenting substrate, so it's essentially a surrogate antigen-presenting cell. So for those of you who'd seen some of the earlier talks, you'll recall the work from Avi Kupfer demonstrating this formation of supramolecular activation complexes. The imaging technology that was used for this was basically wide-field fluorescence microscopy, optical sections through the cell-cell conjugate, and then a computational deconvolution method to reconstruct the three-dimensional structure with greater resolution than would be possible, or at least greater clarity than would be possible without the deconvolution. And it revealed this segregation of the T-cell receptor, shown in green, from the adhesive interactions with these two domains. So this is an example of combining the the adhesion system and the antigen recognition process as a normal cell-cell couple would do. We could also then do this in the supported planar bilayer system. And again, we saw something. So I guess in the movie on your left, basically in the center there, we're basically seeing the T-cell receptor MHC peptide complex interaction in green, the LFA1 adhesion interaction with ICAM1 in red, and essentially what you're seeing is the formation of the pattern that Kupfer saw, this bullseye-like pattern. So this was sort of, it recapitulated what Kupfer had seen and allowed us to see the dynamics. That was very nice. At the same time, we had other synapses form, which were somewhat disordered in the early phases, and then kind of completely decomposed, like the cell actually moved on and did not form a stable junction, did not form the symmetric bullseye-like pattern. But as you can see, what's happening, the red signal, which is the adhesion signal, is actually moving off away from the cluster of T-cell receptor and MHC peptide complexes. And you can see as the cell moves away, based on the red signal basically moving out of the frame, these clusters of MHC peptide complexes with presumably engaged by T-cell receptors are left behind in the substrate. So again, another indication that T-cells were leaving receptor behind with the antigen-presenting cell, which is something that, again, we hadn't expected. We'd expected internalization and degradation of the T-cell receptor to be the normal way to resolve these interactions. So this got us, so there were another observation related to this is if you look at the relationship between the density of MHC peptide complexes in the substrate and the size of the SMAC, so this is basically increasing MHC peptide density in the planar bilayers, and the signal here is basically the MHC peptide complexes. This is the T-cell receptor. So you can see there's a very good correspondence between these, and the intensity of this central structure, SMAC, as described by Kupfer, is essentially linear with antigen increase, and that's kind of quantified here. So in some ways, this finding in itself was also surprising, because if you look at the signaling based on calcium mobilization in the T-cell, it basically is fully activated at around 1 molecule per micron squared, and basically then is saturated at a few molecules per micron squared, and then doesn't increase further. Whereas this basically signal that we were seeing in the SMAC, which initially we thought might be involved in signaling, was completely linear rather than having this threshold-like behavior. So again, this linearity with antigen input was an interesting characteristic that was very at odds with the behavior that the T cell would be displaying in terms of signaling. So again, it left us with a question as to what the purpose of the central accumulation of T cell receptor in the immunological synapse actually is, which initially we thought we had a hypothesis. We thought it was related to signaling, but that seems to be incorrect. So we had to think further about this. So one of the questions that we could then ask is, what is the molecular machinery that creates this central cluster? And a key experiment in understanding this was initiated by Santosh Verdana, who was working in my group at New York University. And what he did basically was knock down a component of a machinery referred to as the endosomal sorting complexes required for transport, or ESCRT. And in fact, there are 3 major complexes in the ESCRT machinery, maybe 4 if you count ESCRT 0, 1, 2, and 3. And this was basically, he's knocking down ESCRT I, which is a protein in mammals referred to as a TSG101, or transforming tumor suppressor gene 101. And essentially, what he found was that when he knocked this down in T cells, he got 2 major phenotypes. One was with the control siRNA, you have a SMAC formation. With the TSG101 siRNA, you basically have this ring of T cell receptor with essentially very little T cell receptor in that central region, in the SMAC region. So failure to form the SMAC. And also, if you look at the level of tyrosine phosphorylation, again, the T cell receptor is linked to a tyrosine kinase cascade. You have this is the physiological level of signaling in the immunological synapse through T cell receptor clusters. This is what we're seeing essentially with TSG101 knockdown, which is about a 10-fold increase in tyrosine phosphorylation. So you had a hyper signaling through the T cell receptor and failure to form the SMAC. So in some respects, this phenotype with the ESCRT machinery fit with some other things that were known about the way this ESCRT machinery deals with growth factor receptors in terms of signal termination. And but the next step in that process, which would be degradation in lysosomes, clearly was not happening in this particular context in the immunological synapse. We were ending up instead with a transport process, which was resulting in the formation of a SMAC when this system was functioning normally. And this was, again, not an expected phenotype for this ESCRT machinery, which would otherwise not be involved usually in sorting out molecules at the cell surface. So again, we need to think about this a little bit more. So just to kind of introduce you to the ESCRT machinery a little bit more, again, I mentioned these basically 4 complexes. We're looking at TSG101 here. And what TSG101 is doing is recognizing ubiquitinated cell surface receptors, which are recognized usually in endocytic compartments and results in, this is the cytoplasmic side, this is the extracellular side, so this would say be the extracellular part of the T cell receptor, this would be the cytoplasmic side on this side, and you're essentially looking at the formation of a bud into the lumen of an endosome, which would result in the degradation of that protein after the endosome fused to a lysosome. So this system is also used for viral budding, though, and that for T cells can happen certainly at the plasma membrane, like in the context of human immunodeficiency virus, which buds from the plasma membrane in T cells. So we became kind of interested in the idea that perhaps TSG101 in this situation is acting with the T cell receptor the way it would with, say, viral glycoproteins in the formation of HIV particles and actually creating a bud into the extracellular space. So to test that, we basically needed to use another set of methods. Now, another observation that we had that essentially fit with this was work from Rajat Varma published in 2006, showed that in some contexts we would see T cell receptor positive particles outside of the immunological synapse that were basically interacting with the supported lipid bilayer and essentially diffusing around in 2 dimensions on that surface. So this again fit with the idea that although many of the T cell receptors appeared to be confined within the SMAC region, under certain conditions a lot of these receptors seem to be getting loose outside of the cell in the extracellular space and at the same time continue to interact with the supported bilayer where the MHC molecules are. So again, fitting with potentially a budding process that would regenerate T cell receptor positive particles that were actually in some cases then moving out of the SMAC region in this model system where there's no antigen presenting cell that could further process those. So to test this, we started using this electron microscopy approach with David Stokes at NYU, and Kaushik Chaudhuri in my group, and Jaime Lodra in David's lab, basically, along with Lance Kam at Columbia, set up a system where they could form supported planar bilayers on a grid, chrome grid, which would basically give us an imprint in the electron microscopy experiment that we could then use to orient our fluorescence imaging shown here to electron micrographs acquired after fixation and processing of the sample for electron microscopy. And due to the skills of a very talented technician who could take these en face sections off the face of these blocks, which would basically be, in this case, maybe 100 nanometer or so section, essentially lined up manually by eye, which is quite a significant skill, we were able to basically correlate, obtain these correlative images where you have in this case, an actin signal that shows you where the cell is, the T-cell receptor signal that shows you where the SMAC is, again, using Kupfer's nomenclature. And essentially, when you look at the electron micrograph from that overlay in more detail, just focusing in on this region where the T-cell receptors accumulated, what you'd see was basically these little rings, these little circles in any kind of level in the tomograms or in the thin sections. And the variability of the diameter of these circles is consistent with that small vesicles. And in fact, serial tomograms that were taken on the vertical axis through the interface, and this is basically just a movie that shows the results of 4 of these tomograms, indicated that you have small vesicles on the outside of the cell. So this line down here at the bottom is the supported planar bilayer. This is the plasma membrane is here, and the structures in the interface between the T cell and the planar bilayer are these extracellular vesicles. And because this tomogram allows these 3-dimensional reconstructions, and that's basically the end of the 4th tomogram, that information is a little bit hard to digest. So Kaushik spent quite a bit of time going in and tracing the relevant structures, the vesicles, and some of the cytoplasmic organelles, and generating this model, which we can then play back as a movie, and basically makes it a little bit easier to focus on the various elements that we found of interest. So typically, immunological synapses have a high degree of cellular polarity. You have the centrioles or microtubule organizing centers in the indicated here in purple, microtubules in lighter purple. The Golgi apparatus is basically up here, and some of these other structures there, endosomes, mitochondria in kind of orange. The blue structure up here is a nuclear envelope. And again, the gold structures in the interface between the supported planar bilayer in light blue and the plasma membrane in green are these extracellular vesicles that are being released into this SMAC structure. Again, quite an unexpected finding to see this really almost complete correlation of the T-cell receptor signal with these extracellular vesicles, which before we had thought basically were T-cell receptors that were still in the T-cell plasma membrane interacting with these MHC peptide complexes on the antigen-presenting cell. But this explained a lot to us in terms of how we would see termination of signaling and this prior evidence of the T-cell receptor being left behind on the substrate, because the T-cell receptor in these vesicles is no longer coupled to the cells. It's now released to this surrogate antigen-presenting cell, but because the planar bilayer is a passive system, it basically can't do anything with them. So what we see when we basically then go back to our cell-cell conjugation systems with T cells and B cells and look at what's happening in the interface between those cells, we can actually see evidence of transfer of T cell receptor into the B cell, which becomes particularly visible. So here's the T cell and here's the B cell, and basically after around 15-20 minutes, we'll see these T cell receptor positive puncta within the B cell, and this is inhibited when we don't have antigen, so there's basically it's antigen dependent, and we can also show that if we knock down TSG101 in the T cell, we can also prevent this transfer. And furthermore, if you look by electron microscopy in situations where the cytoplasm of the T cell is labeled with this, in this case, this kind of silver-enhanced gold labeling, these dark particles that are in the T cell cytoplasm, the B cell in this case, which is here, doesn't have these, doesn't basically have that genetically encoded tag that basically lets us tell the T cell from the B cell. But we see within the B cell vesicles that have double-walled, essentially compartments within the B cell that contain T cell cytoplasm, suggesting that the B cells take these particles up from the T cell, during the process of immunological synapse formation. And this was a form of information transfer that we hadn't previously appreciated. So what we think is happening here is that you have these complexes of T-cell receptor and MHC peptide complexes at the interface between the T-cell and the antigen-presenting cell. And our suspicion is that these complexes, when you have enough MHC peptide complexes, become effectively irreversible. And certainly B cells can take up a lot of antigen due to their high affinity B cell receptor to concentrate the antigen. So we think these are the situations where you'd have these high density MHC peptide complexes that create these essentially irreversible interactions. And then the T cell can either, the system between the T cell and the B cell have to negotiate then what to do with this for them to be able to separate. So in this situation, we think there are two ways you can go. One way basically is for the T cell to essentially pull off a patch of membrane from the B cell through an endocytic process where it would basically use molecules like clathrin and dynamin to essentially create this invagination and pull off a bit of the B cell membrane with the MHC peptide complexes. Or it can basically go the other direction and it can use the ESCRT machinery to release a vesicle towards the B cell using the ESCRT I proteins like TSG101 to initiate the sorting, the ESCRT III polymers to basically create the vesicle, and then VPS4, this ATPase, to actually scission the vesicle off and freely release it to the B cell, which can then internalize that fragment from the T cell. And we have evidence in looking at the T cell B cell conjugates for both phenomena happening. So basically both T cells transferring vesicles to the B cells and pulling vesicles off the B cells. So we're still studying basically what regulates the balance between those two phenomena, but there's definitely always a prominent component of this ESCRT-dependent vesicle release. So as I showed you in those very early movies of the immunological synapse on the planar bilayer system, T cells, when they break symmetry and leave a particular site, will leave behind trails of T cell receptor interacting with MHC peptide complexes. And we can also see this in the electron micrographs as the release of trails of vesicles that the correlative light and electron microscopy shows are red or T cell receptor rich. And basically have an example here and kind of a blow up here. So this is a very common phenomenon in our preparations and gave us the opportunity to basically then determine what happens if we take B cells with appropriate MHC peptide complexes and bring them into contact with these trails of T cell receptor positive vesicles that were released by the T cells. And what we see in this context is signaling in the B cell. So if the B cell basically with the appropriate MHC peptide complexes engages T cell receptors on these released vesicles, we see an increase in cytoplasmic calcium in the B cells, suggesting the B cells are getting activated by the vesicles distinct from any interaction with the T cells. So one idea would be that T cells could transfer material to the B cells. And in fact, in vitro systems, we can quantify this transfer by a method like flow cytometry, where we can basically look at the T cell receptor fluorescence signal on this axis and essentially see that based on gating on B cells in the other axis, that we can see B cells with increasing amounts of MHC peptide complexes when we increase the level of essentially surrogate antigen molecules, in this case so-called staphylococcal superantigen, that bridges the T cell receptor and the MHC peptide complexes and allows us to experimentally essentially dose in higher levels of T cell receptor engagement, which generates transfer of T cell receptors to the B cells. And in fact, other signals seem to be moving through a similar conduit. And we see molecules like CD40 ligand, which is an important non-antigen specific signal that can increase the proliferation of B cells and their differentiation, really a critical signal in T cell B cell communication. So the molecule itself is not antigen specific. It's essentially like a cytokine, but its specificity comes from the fact that it's being conveyed through this immunological synapse, and we think it's being conveyed in the form of these vesicles released along with the T cell receptor to the B cell. So this would help us understand how the T cell receptor positive vesicles and their transfer could be linked to other signals the B cells need to differentiate, and for example, to undergo processes like antibody class switching to make the appropriate type of antibody, and affinity maturation to make higher affinity antibodies in processes in lymph nodes. And one model that we have, or hypothesis that we have for this, is that the transfer of these vesicles into a B cell could help distinguish high affinity and low affinity B cells based on the high affinity B cells or B cells with high affinity antigen receptors collecting more antigen, making higher densities of MHC peptide complexes, B cells with lower affinity B cell receptors making lower numbers of MHC peptide complexes, and then essentially the amount of the MHC peptide complex is then being translated into a number of these vesicles, and B cells that basically collected more of these vesicles through having more MHC peptide complexes would basically then undergo more rounds of division, and this would then, based on the uptake and effectively serial dilution of these vesicles from the T cell, would be able to effectively count the number of times that they would divide before they would stop dividing, and then could potentially differentiate into antibody-producing cells or further mutate their receptor for more rounds of selection. So this would be kind of a bit of a token economy that the T cell would be using to basically convey to B cells they want to help an ability to undergo a larger proliferative burst and compete better in this process of making high-affinity antibodies. And then the low-affinity B cells would be less well rewarded and would proliferate less and eventually could be outcompeted by such a process, allowing the higher affinity cells to essentially dominate the response. So in conclusion, we have discovered that there are these T cell receptor enriched extracellular vesicles that are generated in the immunological synapse and effectively make up a large part of the SMAC originally described by Kupfer, and that could be recapitulated in the supported planar bilayer-based reconstitution experiments that I've shown you earlier. ESCRT machinery, including TSG101, is required to terminate signaling and sort these T cell receptors into these vesicles, which then are released to the B cell. And this VPS4 protein can be shown to be critical for the actual scission of the vesicles and the final transfer. So these microvesicles activate B cells in an antigen-dependent manner, and we've also found critical signals like CD40 ligand mixed in with these vesicles in the SMAC, and this may suggest that this vesicle transfer process may have a critical role in T cell health for B cells. And this was something that really we feel that was discovered through this reconstitution approach and really kind of demonstrates the value of some kind of biochemical approaches when applied to live cells like the T cell immune system. So there are a number of colleagues who basically contributed to this work, both at NYU, Harvard Medical School, Columbia. I basically mentioned all of the names during the presentation, but I just want to leave them up here, and also want to thank the National Institutes of Health and the Wellcome Trust and Kennedy Trust in the UK for supporting our work in this area. Thank you.